The next topic is fatty acid biosynthesis. Fatty acid synthesis is not the reversal pathway of fatty acid oxidation. This fatty acid synthesis takes place in cytosol. The starting material of fatty acid synthesis is acetyl-CoA, and the synthesis always ends in the formation of palmitic acid. The materials required for the synthesis are the enzymes fatty acid synthase complex and acetyl-CoA carboxylase. The manganese and bicarbonate acts as a source of carbon dioxide, and ATP is a source of energy. An important multi-enzyme complex is going to participate in biosynthesis. This is fatty acid synthase complex. In yeast, mammals and birds, the synthase enzyme system is called as fatty acid synthase complex. It is a multi-enzyme complex. Each monomeric unit contains 6 enzymes and an ACP complex. It is actually a dimer with 2 identical polypeptide monomeric units. The ACP has an sulfhydryl group, that is SH group, in the 4-phosphopantathene moiety referred as pantothenol SH or PAN-SH. Another active sulfhydryl group present in the cysteine moiety of the enzyme ketoacyl synthase referred as cystenyl SH or cis-SH. The PAN-SH of one monomeric unit is in close proximity to the cis-SH group of another monomeric unit and vice versa. And I am going to tell you the order of enzymes from end to end in each monomeric unit. 3-ketoacyl synthase, trans-acylase, enoyl reductase, 3-hydroxyacyl dehydratase, 3-ketoacyl reductase, ACP or acyl carrier protein, and thioesterase. This is the order of enzymes from end to end. It is found that complex is functional only when the 2 monomeric units are in association with each other. The functional activity is lost when they are dissociated. In a dimer form, the complex jointly synthesizes 2 molecules of palmitic acid simultaneously. Let us move on to the steps of fatty acid synthesis. The starting material for the synthesis of fatty acids is acetyl-CoA. Acetyl-CoA is formed in mitochondrion, but synthesis occurs in cytosol. Acetyl-CoA is impermeable to mitochondrial membrane, and various steps are going to occur in cytosol for the formation of palmitic acid from acetyl-CoA. We will discuss how. Let me explain you the steps involved in fatty acid biosynthesis. The first step is formation of malonyl-CoA from acetyl-CoA. In the presence of the enzyme acetyl-CoA carboxylase, the acetyl-CoA is converted to malonyl-CoA by carbon dioxide fixation reaction. Manganese is required. The reaction is irreversible and carbon dioxide is provided by bicarbonate. One high energy bond of ATP is utilized. Acetyl-CoA carboxylase is the rate limiting enzyme, and citrate is an activator of the enzyme, and palmityl-CoA is inhibitor. Once malonyl-CoA is formed from acetyl-CoA, rest of the fatty acid synthesis reactions take place with fatty acid synthase complex. cis-SH and pan-SH may be considered as two arms of the enzyme complex. Cis-SH is the acceptor of acetyl-CoA, whereas pan-SH takes up malonyl-CoA. Initially, a molecule of acetyl-CoA combines with cis-SH of ketoacyl synthase of one monomeric unit. The coenzyme A is removed. The reaction is catalyzed by the enzyme transacylase. In a similar manner, a molecule of malonyl-CoA combines with the adjacent pan-SH of ACP of opposite monomeric unit to form malonyl ACP enzyme. The coenzyme A of malonyl-CoA is also removed in this step, and the reaction is catalyzed by the enzyme transacylase. Now, a condensation reaction is going to occur in which the acetate attacks malonate to form acetoacetyl ACP. The reaction is catalyzed by the enzyme ketoacyl synthase, which is also called as condensing enzyme, and there is loss of one molecule of carbon dioxide. So this decarboxylation reaction provides the extra thermodynamic push to make the reaction highly favorable. The acetoacetate remains attached to pan-SH of monomer 2. The cis-SH of monomer 1 becomes free. While acetoacetate remains attached to pan-SH, three reactions take place, that is reduction, dehydration, followed by another reduction. The first reaction called reduction, in which the ketoacyl group is reduced to hydroxyl group to form beta-hydroxybutyl ACP, catalyzed by the enzyme ketoacyl reductase. The second reaction is dehydration, in which a molecule of water is removed from beta-hydroxybutyl ACP to form alpha beta unsaturated butyryl ACP. This is catalyzed by the enzyme beta-hydroxyacyl dehydratase. The third reaction, called as reduction reaction, which is the final reaction, is catalyzed by enoyl reductase using NADPH and H+. As a result, the double bond is saturated to form butyryl ACP. All the three reactions occur on pan-SH of monomer 2. Once saturated butyric acid is formed, it is now transferred to the cis-SH of monomer 1, which is free to accommodate. Now, a fresh molecule of malonyl-CoA is taken up onto the free pan-SH group of monomer 2, and the sequence of events is repeated to form a saturated 6-carbon fatty acid. Once formed, this is again transferred to cis-SH of monomer 1. The set of reactions on each of the monomer is repeated till a 16-carbon palmityl ACP is formed on pan-SH of monomer 2. Palmityl ACP is released as palmitic acid from the enzyme complex by the enzyme thioesterase. And what is the source of acetyl-CoA, and where is NADPH obtained for this fatty acid biosynthesis? So let us see in detail. The sources of acetyl-CoA is mainly found in mitochondria, which cannot pass out. It forms citrate by condensing with oxaloacetate. Citrate is transported out by a transporter protein in exchange of malate. Once in cytoplasm, an enzyme citrate lyase cleaves citrate with the help of ATP to form acetyl-CoA and oxaloacetate. Carnitine acetyl transferase may probably transfer acetyl group of acetyl-CoA to carnitine to form acyl carnitine in mitochondria. After translocation to cytoplasm, acetyl group may be transferred to CoA to make it acetyl-CoA. What is the source of NADPH then? Hexose monophosphate shunt. It is otherwise called as pentose phosphate pathway, is the main source of NADPH. An enzyme called isocitrate dehydrogenase, which is present in the cytoplasm, can generate NADPH mainly in ruminants. Coming to the regulation of fatty acid synthesis, so it can be regulated in two different ways. The activities of enzymes involved in fatty acid synthesis can be controlled by short-term control and long-term control. Short-term control, which involves allosteric or metabolic regulation and covalent modification of enzymes. In long-term control, this involves changes in the amounts of enzymes brought about by the changes in the rates of synthesis and degradation. Primarily, acetyl-CoA carboxylase catalyzes the rate-limiting step in the de novo synthesis of fatty acids. It is also believed that regulation of acetyl-CoA carboxylase is done by phosphorylation and dephosphorylation of the protein. Fatty acid synthesis may also be regulated by altering the activity of pyruvate dehydrogenase, which oxidatively decarboxylates pyruvate to acetyl-CoA. Long-term regulation is done by stimulation of acetyl-CoA carboxylase, fatty acid synthase and ATP citrate lyase. Insulin increases fatty acid synthesis in several ways, such as decreasing lipolysis, bringing about activation of protein phosphatase, stimulating synthesis of citrate lyase, enhancing formation of acetyl-CoA from pyruvate, increasing glycolysis, which leads to increased pyruvate and thus acetyl-CoA. So with this, we have completed discussing about fatty acid oxidation and fatty acid biosynthesis.